step by step, we're going to have a look at monoclonal antibodies. In the news a lot at the moment because it could be the way of trying to save the world from COVID-19. But it also finds a use in other things, and one of those common things is pregnancy testing. So let's have a look at what is a monoclonal antibody and how we go about making them. Now, a monoclonal antibody is used sometimes in vaccinations, so monoclonal antibody, so we've got use in vaccinations and used largely in testing, and for things like the COVID-19 outbreak, testing for the coronavirus and testing to see whether you've got those antibodies is really quite important. Simply, an antibody is a protein, and we've got to try and make a protein. Now, how do we go about doing that? Well, we've got to try and produce those proteins. So, some special white blood cells known as lymphocytes, they make the antibodies, but those cells can't divide. We might find we've got some tumour cells which don't make the antibodies, but they can divide rapidly to make lots of clone cells. All mammals, including mice, make these lymphocytes, and scientists can combine these mice lymphocytes that are now stimulated to make one particular antibody with a type of tumour cell to make a cell called a hybridoma. And this hybridoma basically identical cells that can all produce antibodies. So, how do we go about doing this? Well, in very simple terms, we start off with my favourite little animal. There we are. Don't look much like a mouse, does it? Right, so we take our mouse and we take some of these tumour cells. We combine them to make a hybridoma cell. This then can divide and divide and divide and divide, and each of these cells is producing these little antibodies. Now we can basically collect these antibodies, we can purify them, and then we can basically use them for all types of purposes. Now, one of those might be a pregnancy test. So let's have a look at this. A typical pregnancy test might look something like this, and we have one line if you're not pregnant and maybe two lines if you are. The colour can change, that's not important, and they can, if they get really desperate, even write in words like pregnant or not pregnant to try and help people to see whether they are or not. Try and make these things easier. So let's see how we go about doing this. So we know that the monoclonal antibodies are antibodies from a particular cell. Now, trying to identify that cell is reasonably difficult. So what we could do is we could take our mouse again, and what we could do with this mouse is we could inject this mouse with a particular chemical, and this chemical is called HCG. HCG here is a hormone produced by a woman when she is pregnant. Human chorionic gonadotrophin. Now, some of this hormone is found in the blood and in the urine of the mother when she becomes pregnant. So what we can try and do is we can put some of this hormone into this mouse, and this mouse will produce a particular antibody to this because it's a foreign substance to this particular mouse. Now, these are made in these lymphocytes. We'll call them B cells, which is their proper name. And what we can do then is we can basically extract those from somewhere like the spleen of a mouse. As you can gather, this isn't going to do the mouse an awful lot of good. Once we've got those cells, they're fused with this cancer cell, this myeloma, a type of cancer cell that divides very rapidly. Now, as things go on, nowadays we're trying to also join these to perhaps some human cells, which are making them even more of a weird hybrid. And it's something that I suppose that worries people, because, you know, imagining trying to combine cells from mice and people and cancer cells to form a new cell sounds a little bit horrific, perhaps far-fetched, perhaps even dangerous. However, we're finding that these cells, these hybridomas, very useful in trying to make these cells which can now produce antibodies for you. So we've got those, and we're going to join them up. So we've got this system now where we're making lots of these antibodies. Now, what are we going to try and do? Well, we've got to try to sort of identify what's going on. So if someone is pregnant, so let's take our pregnant person. Apologies to all people for my drawing. Now, what we can do here is we could take a sample here of urine, and that we're going to put onto basically a wick, I suppose. It's a piece of substance that's going to actually absorb some of this urine, and also we hope this HCG, human chorionic gonadotrophin, and we're going to take that, and what we're going to do with that is we're going to put it with some of these antibodies. The antibody, so if we draw out this little hormone, HCG, and this hopefully will bind to this particular antibody. Now, if the woman's pregnant, it binds to these, and these things then could combine and join to something with hopefully some sort of coloured bead next to it. And let's see if we can sort of get the idea of what's going to go on here. So if we try and have a look at what one of these tests looks like, we've got this sort of picture. Let's see if we can draw what's going on inside of that, and hopefully we can get some ideas. So let's draw out this. I've got a little window, and I've got some basically immobilized monoclonal antibodies which are used to bind to mobile antibodies. So let's put some in here, and what we've got is perhaps something like that, and we'll stick on here. So what we've got going along is this mobile antibody, and this binds to the immobilized monoclonal antibody to produce something. So any of those accessed here, and they form then a coloured line whether the woman is basically pregnant or not. So these excess mobile antibodies can join on. Now let's look at another one. Now, if we've got this system here, and we've got our monoclonal antibody here, and this is going to bind to our human chorionic gonadotrophin, and we can put it here. This combines with this, and this produces here basically a coloured line if the woman is pregnant. So let's start this process. Here is our wick. It's got urine in it, and it's got some human chorionic gonadotrophin in it. In here we've got a monoclonal antibody, and this will combine with that. Now, some of those molecules will be up here. If this works, then it binds to this and we get a colour. If it isn't, these will bind to those only and they'll get a colour. So if we get 2 bars here, you're pregnant, so this is, if you like, the pregnant one, and this one is just collecting all the antibodies, so this one is your not pregnant. So if you get 1 line, you're not pregnant, and if you get 2 lines, you are. So that's sort of trying to deal with something like pregnancy. Works in the same way with not just pregnancy, but here diagnosis of a disease. And I suppose we're looking at COVID-19 here. Right, so let's see how we might try to do this. So we make some monoclonal antibodies which will bind to something on the virus. So I've got my virus, it's got various chemicals on the outside, and we can make one of these bind to this. This is not as simple as it sounds, but the human body will produce these things. So we may have these monoclonal antibodies. Now, what we would need to do is to ramp up the production of these, so once we've got one we can make a whole load of these. And once we've done that, we need to stick this to some sort of colour. So that this may bind to something else that we've got on a stick. And we're going to try to do this to make sure that this will stick to this if perhaps we've got some of these antigens present. So if these antibodies, in this case, you've made some antibodies, you've got an antibody here which is picking up the antibody, and if it is, then it will stick to something and it will change colour. So we can see how these work. Now these are basically very sensitive. In fact, they are so sensitive they work on just a few molecules, so they're pretty good here. One of the other uses of this is for men with things like prostate cancer. If a man's got prostate cancer, prostate gland, very hard to sort of spot because it's inside the body, very hard to try and work out if something's gone wrong with it. But if this is producing something that we can then detect in a blood test, then we can get perhaps one of these chemicals in here, one of these antibodies, antigens, to bind to a colour, and we can see this, and you can see then whether they've got this disease or not. So we're trying to sort of bind these antibodies that will bind to either COVID-19 or something else in the system, so we can identify what's going on. We can use all this for various other basic purposes. So we've got pregnancy testing, diagnosis of diseases, but we can use this for all sorts of measuring. We can measure levels of hormones, chemicals in the body. You may have heard of things like clinistics, which are sticks that will go into urine, and they can then be used to measure all types of things. One of them being diabetes. We can use it largely in research to try and identify, locate specific molecules in a cell or a tissue, and we can often attach these to some sort of dye. Fluorescent dyes quite often used, so we can see what's going on, and we might be able to even use it to treat a disease. So let's imagine we want to try and treat a disease, and one of these could easily be something like cancer. So we want to try and beat cancer. Not as easy as it sounds, because cancer isn't one sort of cell. We don't have a disease called cancer, but there are 200 or more different types of cancer, each one different. And what scientists have been looking for is something called a magic bullet, one chemical that could perhaps just bind to all cancer cells and destroy them. It'd be wonderful, but it doesn't quite work out quite as simply as that. So we know that a monoclonal antibody is useful for basically binding to a chemical. So how about trying to treat a disease with this? So let's suppose I've got one of your cells and it's gone cancerous. It's a cancer cell. Now all that means is this cell should reproduce and then stop. But it's cancerous. It now is just reproducing and reproducing and reproducing, and it's making so many of these cells that it can cause blockages. On its surface, this cell will have a collection of markers, an antigen, and what we want to do is we want to try and produce some sort of monoclonal antibody so that it will stick to this cancer cell and only this cancer cell. And these little things send out little sort of messages, and they send these out to some sort of white blood cell, and they say, come and destroy me. Now, what are the problems here? Well, let's see if we could identify a cancer cell. So the first question I suppose that comes up is, does a cancer cell have basically a different antigen? If the answer to this is yes, then we can make for this a monoclonal antibody. And perhaps to this monoclonal antibody, which typically looks like a Y-shaped molecule, maybe I could in fact even attach something which is sort of radioactive. This would put a radioactive source right next to this cancer cell, which certainly wouldn't do it an awful lot of good and would kill it. Or we could use it just to send out a message to tell other white blood cells to come along and just destroy this. So these monoclonal antibodies here will basically now block these receptors on the surface of the cell, and that might actually also just stop it growing. And that would work really just as well. We could put, as I said, radioactive chemicals there. I could also put something that's fairly toxic, a toxic chemical on there, and that will bind to this particular cancer cell and kill it. And the good news is it would do that without killing any other cell in the body. So monoclonal antibodies could be here the answer to this. And a guy called Paul Ehrlich managed to win a Nobel Prize here for trying to get this idea of this magic bullet, this drug precisely targeted to a particular pathogen or a disease. And when these monoclonal antibodies were first developed in about 1970, late 70s, doctors and scientists thought that maybe this would be the answer. But it's not quite the answer, because we've got with this some certain advantages, but also it comes with some disadvantages. The biggest advantage here that we can think of using monoclonal antibodies is they only bind to some sort of disease, cancer cells, and so on. Any other healthy cells, it doesn't bind to them, so they're not affected at all. Whereas in contrast, conventional drug treatment would affect all cells. So when people have chemotherapy, we are targeting the cells that grow really fast. Now these are the cancer cells, but they're not the only ones. Hair cells are also reproducing, trying to produce all this hair, and they die, so people's hair falls out. So it's got a few problems. Any other fast reproducing cells also are liable to die. And that could affect your immune system as well. So that's not quite so good. We want something that is more specific, that targets just one thing. So we're only targeting these cancer cells. So we've got chemotherapy, radiotherapy. These will target a much wider area. So that's going to affect all sorts of other things. Now, a disadvantage of this is we can imagine that this is expensive. I was looking up how much a coronavirus test kit was going to cost with one of these things, and you know, it was about sort of £500. Now, although this was developed sort of, let's say about 1980, so 40 odd years ago, we've actually found that there are lots of side effects. Partly due to the fact that we're actually using mouse cells, and because we've got a mouse cell forming this hybridoma, then it's got its own difference to a human. And so these cells, when they produce these antibodies now can also trigger an immune response in the human. So you put in these antibodies into a human, and your first job is, rather than using them and helping you, you actually start destroying them because you don't know any better. So this is why we're now trying to take mouse human hybrid cells to try and alleviate these problems. We can try to get some of these monoclonal antibodies to hold things like radioactive source or a toxic chemical, and this will only target one thing. However, attaching drugs to a monoclonal antibody or radioactive sources is difficult and very time consuming. But we're making progress, and maybe at the moment we've got some part of one of these magic bullets, even if we haven't got the whole thing. So monoclonal antibodies are useful, but they do have some problems, and we're working on trying to get those problems solved. Initially we thought one of the big problems was trying to connect it to a dye, a coloured molecule, and that would then bind to something else so we could actually see it. That we solved fairly quickly and easily. Trying to get other chemicals to bind to these monoclonal antibodies, much more difficult. Trying to isolate this monoclonal antibody as well to sort of just an antibody to try and work on proves more difficult than you'd imagine. So these things don't appear overnight. When COVID-19 hit, relatively quickly we got some tests for it, but they weren't reliable. They didn't work every time because they weren't good enough. It takes time to try and get some of these things out. There's a lot of pressure on these drug companies to try and get something out first, faster than anybody else, because if you're first on the market, then everybody's going to buy from you while other people are still trying to develop. But it's got to work. And we had a lot of debate here in England about people saying, especially in the press, so-and-so's produced one of these monoclonal antibodies for a test. And until we knew and we could test it to make sure it was 100% reliable, it wasn't much use. We've got to make sure that these tests are good. They've got to be basically infallible. Pregnancy tests. Someone doesn't want to know they might be pregnant. They either are or they're not, and they want to know whether they are or they aren't. And trying to get one of these systems to work takes time, it takes effort, it takes quite a lot of money and development. And in fairness, these companies need to try and recoup some of that time, effort, because without that, they go bust. So that's something we have to sort of bear in mind when we're trying to complain about why it takes so long to produce some of these chemicals. It is a case of trying to make it, but also test it and make sure that it's basically infallible. So monoclonal antibodies, very useful, but the big problem in monoclonal antibodies is trying to sort of make them, isolate them, check that they work. So we've gone through then the advantages and disadvantages. Cost is still one of the big problems. The side effects are also really one of the big problems, and attaching drugs, something we're solving. The good points, it only affects the one cell, and that's the important bit. If we can only get it to affect one cell, that we're doing really well. If this affects all sorts of cells, then basically it's useless to us. So we think about some of these names. Monoclonal antibodies can be used to make something called enzyme-linked immunoabsorbent assays. That sounded horrible, didn't it? Enzyme-linked immuno just this is fortunately has an abbreviation. These things are used to detect antibodies, pathogens in a sample. They can also be used to detect all sorts of other things. So let's summarize. Monoclonal antibodies are produced from a single clone of some cells. Each type is specific to one particular binding site on some sort of cell, virus, anything you particularly want to look at. They can also target, maybe, specific cells. We use these monoclonal antibodies. They're produced by putting them into a mouse with a tumour cell, and that can then create these monoclonal antibodies, which are collected and purified at great expense to the mouse. And then we can use them in pregnancy tests, various diagnostic level tests to work out levels of hormones, different chemicals in the body, and we can use them to identify specific cells. So we can use them in treatments of diseases. We can use them against certain cancers at the moment. We can bind toxic chemicals, radioactive drugs, all sorts of things that will stop those cells dividing, and then they won't harm any other cells and they won't cause any problems. But they do have lots of side effects, and that's what we're working on at the moment. Well, I hope you found this useful, and if you did, then please subscribe, and I will see you next week when we'll have a look at some more GCSE biology, topic by topic. Until then, take care and stay safe. Bye bye.